meeting. Now, as aptly called by the chairperson, this dry symposium that you're having over here kind of dissuaded the audience, and right from the first presentation I was witnessing a steady decline in the audience population. So I was discussing with the speaker to follow, Dr. Balasubramaniam, Balu as I call him, that by the time you take the podium, maybe there will be hardly anybody left to listen to you. But now it's encouraging to see that we have a sizable audience at the end of the day. Well, to go to the next slide. Yeah. Now, when you have a bacterial infection or an injury, you have an immunological response which is mediated by inflammatory markers. Overnutrition is no exception. The immune system perceives a surfeit of nutrition as a threat to the body, and it mounts an immune response which is mainly of an inflammatory nature, and the immune system tries to restore metabolic homeostasis. So you have an excess of fat, you can tuck them away safely in target tissues like liver and adipose tissues, so you can have a safe storage of fat. The problem starts when you have unsafe storage of fat in liver, in the visceral adipose tissue, and in the skeletal muscles, and you start having all sorts of inflammatory reactions that lead to insulin resistance, metabolic syndrome, and diabetes. So I have taken this cartoon from Nature Drug Discovery, which I find very, very illustrative. You have a normal population of beta cells over there in the pancreas, and you have a normal population of adipose tissue there. Now, if you are overfed, if you are on a calorie-dense diet, mainly a fat-dense diet, your adipocytes undergo hypertrophy. If they are in the safe location, not much of inflammatory reaction. If you have the genetic predisposition to develop truncal obesity, type 2 diabetes, fatty liver disease, you have unsafe storage of fat here, and when those adipocytes that are destined to become inflammatory, they get overloaded with fat, they become inflammatory in nature. And the underlying mechanism is very interesting. When you have unsafe storage of fat in adipose tissue, adipocytes undergo hypertrophy, and the vasculature that is essential for maintaining nutrition to those adipocytes cannot keep pace with the rate at which the adipocytes undergo hypertrophy. So there is a relative vascular insufficiency, hypoxia, and that leads to generation of oxidative stress, and the adipocytes become inflammatory in nature. So they invite your macrophages and T cells. You can see nice crown-like structures, and following T cell and macrophage infiltration, the adipocytes become resistant to insulin-mediated glucose uptake. So what happens? A state of insulin resistance develops at the adipose tissue. Now the beta cells, trying to compensate for that insulin resistance, they have to work overtime to generate an increased insulin response. In the process, what happens? The beta cells undergo hypertrophy. And when they undergo hypertrophy, they come under endoplasmic reticulum stress. They undergo hypertrophy, they have to synthesize insulin at an increasing rate, and those insulin molecules are not properly folded to come to the surface. This is known as misfolded protein response. That leads to endoplasmic reticulum stress. Now the beta cells themselves become the target of immune attack, as you can see. Macrophage infiltration that lead to beta cell apoptosis and eventually type 2 diabetes. So like I said before, you can have a safe storage of fat there and nothing much happens. On the other hand, when you have visceral obesity, you have unsafe storage of fat leading to subclinical inflammation. Similarly, in the liver, you can have a benign fatty liver. And those patients do not progress to fatty liver disease, namely NASH and further down. On the other hand, if you have unsafe storage of fat in the liver, so you develop hepatic insulin resistance, hyperglycemia, dysglycemia. So the critical question comes, how can you identify unsafe storage of fat in the liver, in the adipose tissue? Do you have markers? Well, as far as the liver is concerned, when you have unsafe storage of fat in the liver, you have several hepatokines in the circulation. Increased synthesis and release of multiple hepatokines, as many as 6 have been described, and most of them lead to insulin resistance, inflammation, not only in the liver but also in the adipose tissue and skeletal muscle. Now, out of the 6 hepatokines, we have experience with the selenoprotein P and fetchwin A, so I will restrict my subsequent set of slides only to those 2 hepatokines. Selenoprotein P, as you all know, is a selenium-carrying protein. It's a bona fide hepatokine, but it's being recently implicated in insulin resistance. So we undertook a small cross-sectional study to look at the correlation between selenoprotein, insulin resistance, and inflammatory markers. After screening 1,266 individuals from camps organized for people with diabetes in the community, we ultimately could pick up 64 pre-diabetic individuals and 32 diabetic individuals. So we've had a study population comprising of 136 individuals, 64 pre-diabetics, 38 type 2 diabetics, and 34 with normal glucose tolerance. In this small study, what we found was that selenoprotein had a strong inverse correlation with insulin sensitivity as measured by Quickie. You can see over here very strong correlation coefficient even in NGT. So NGT pre-DM type 2 diabetes, very strong negative correlation between selenoprotein P and insulin sensitivity. Now people asked us why NGT individuals have such a strong negative correlation. Well, my answer to that is that as Asian Indians, we are highly insulin resistant, and so such a high correlation between the two, even in normal glucose tolerance individuals, suggests that intrinsically, genetically, we are hepatically insulin resistant, and that's why we have such a strong correlation even in normal glucose tolerance. And when you looked at the correlation between selenoprotein P and inflammatory markers, the study population was small in size, like I told you at the beginning. So we had a weak positive correlation at that point of time when we presented this data. So selenoprotein P appears to be a good predictor of insulin sensitivity and hepatic insulin resistance. Further studies, of course, are needed to find out causality. And our cohort is currently under follow-up. We have also increased the sample size in between, and we are now finding stronger correlation between selenoprotein P and inflammatory markers in pre-diabetic and diabetic population. So we presented this paper in the recently concluded Endocrine Society's 97th annual meeting at San Diego, California. So selenoprotein P is a predictive of insulin resistance and systemic inflammation in individuals with prediabetes and diabetes. Now let me switch gear from here and pass on to a more fascinating hepatokine with which we have made many original contributions in the last 5 years. So this is a blown up view of a hepatocyte where you can see that both glucose and saturated fatty acids, they stimulate the production of this hepatokine pro-inflammatory nature called fetchwin A, albeit through different mechanisms. Saturated fatty acids work through NF-kappa B, while glucose works through the ARP1 and 2 pathway. But no matter how the synthesis and release are triggered, once you have an excess of H1A in the circulation, it induces insulin resistance by two major mechanisms. First of all, it's a direct and potent inhibitor of insulin receptor tyrosine kinase in the target tissues, mainly muscle and adipocytes. And secondly, and more interestingly, H1A in combination with free fatty acids form a dimer which then goes on to and bind to toll-like receptor 4 present on the surface of the immune cells and adipocytes. In fact, this part of the story, which is reviewed by Stephen in Nature Reviews Drug Discovery recently, is based largely on our original work over the last 5 years. So our group was the first to show that the saturated fatty acids do not stimulate the TLR4 directly. You need an adapter protein in between to form a trimer, and it's H1A which acts as that adapter protein and induces an inflammatory response in the immune cells and in the adipose tissue. So this was the first publication where we showed that lipids induce the production of H1A from the liver, and this is actually mediated by NF-kappa B. When you have excess of H1A coming out of the hepatic tissue, it induces adipocyte dysfunction and thereby giving rise to insulin resistance. So in this mice model here, you can see the DB mice, they are naturally obese, and you have the high fat diet mice over here, massively obese and insulin resistant. Now they have a high level of free fatty acid in the circulation, and you can see whenever they have a high FFA in the circulation, there is a parallel increase in H1A protein levels on immunoblot. So a direct link between FFA level in the circulation and production of H1A. And then when you have insulin resistant sera from insulin resistant humans and mice models with a rise in serum levels of H1A, you see an increase in the protein levels of TLR4 and phosphorylated NF-kappa B. So there is a clear link between elevated H1A and those pro-inflammatory cytokines, pro-inflammatory response, NF-kappa B and TLR4. Now high H1A coming out of the liver inhibits adipocyte function. So in this elegant experiment, actually this is a simple experiment called oil red O, where what we did was we treated human adipocytes and mice adipocytes with adipocyte differentiation media or the nutrient media. One of the primary functions of adipocytes is to take up lipids, and when they take up lipids, you have this nice little red droplet signifying normal lipid uptake by the adipocytes on exposure to the nutrient medium. Now see what happens. The moment you add H1A here, there is complete abrogation of lipid uptake by those adipocytes. Now you all know that rosiglitazone and pioglitazone, they are powerful insulin sensitizers, and by themselves they improve lipid uptake by the adipocytes, as is shown over here. The moment you add H1A, so powerful is its action on inhibition of lipid uptake that it even overrides the action of rosiglitazone and pioglitazone. So completely inhibits lipid uptake by those adipocytes. And when you have this pro-inflammatory response leading to impaired lipid uptake, at the same time you have a significant elevation in the plasma levels of the three most important pro-inflammatory cytokines, interleukin-6, TNF-alpha, and IL-1 beta. The next most important function of an adipocyte in terms of metabolic effects is glucose uptake. And see what happens here. When you add the nutrient media here, you have normal expression of the PPAR gamma, the adiponectin level goes up, and all the inflammatory markers are down. Then when you add H1A here, which acts best in presence of a small amount of calcium in the media, what you observe is a complete separation of PPAR alpha, adiponectin, so all bad things start happening, and you have elevation in the levels of pro-inflammatory cytokines. So a powerful pro-inflammatory response there. And when you have such a pro-inflammatory state on hand, what happens is glucose uptake suffers. So glucose uptake by the adipocytes, is dependent on glucose transporters called glucose transporters 4 or GLUT4 that are present inside the intracytoplasmic vesicles in the resting state. The moment you add insulin, insulin translocates those transporters on the cell surface, so now they are in a position to mop up glucose. That's how glucose uptake, insulin-mediated glucose uptake takes place in the adipocyte and in the skeletal muscle. See what happens when you add Phe2 A. Even in presence of significant amount of insulin, Phe2 A completely abrogates insulin-mediated glucose uptake, insulin-mediated translocation of GLUT4 to the cell surface. They're all sitting idle inside the cytoplasmic vesicles. The cell is not in a position to take up any glucose from the surroundings. So that's how Phe2 A inhibits glucose uptake and leads to hyperglycemia. So here you have the two pathways. This is the pathway to show how Phe2 A acts as an adapter protein between FFA and TLR4 and mounts an inflammatory response. The previous speaker told you about lipopolysaccharides, bacterial LPS, also causing insulin resistance. The problem is when you try to find an anti-inflammatory approaches to manage insulin resistance, you can hardly tinker with this pathway because the LPS pathway is intimately linked to innate immunity and can lead to significant immune suppression. So here you have an opportunity with the identification, isolation and characterization of the mechanism by which Phe2 A causes insulin resistance by our group. You can have interventions in place to block here, NF-kappa B or the IK beta, and in fact several clinical trials are currently underway. They are trying to block these pathways without affecting the innate immune response pathway to improve insulin resistance. So to cut a long story short, free fatty acids stimulate the production of Phe2 A from the liver, to which it binds to form a dimer because Phe2 A acts as a carrier protein for free fatty acids. The dimer then goes on to bind to TLR4 present on the surface of immune cells and adipocytes. There is release of pro-inflammatory cytokines mediated by NF-kappa B, which ultimately leads to insulin resistance. So this work of ours got published recently in Nature Medicine as Phe2 A acts as an endogenous ligand of TLR4 to promote lipid-induced insulin resistance. And Jerry Olewski and his group, while writing the editorial on our paper, they commented that Phe2 A is a missing link in lipid-induced inflammation, and Phe2 A is now shown to act as an adapter protein between FFA and TLR4, providing the missing link between free fatty acids and chronic low-grade inflammation that impairs sensitivity. Jerry went on further to suggest that given that most anti-inflammatory approaches to prevent insulin resistance may lead to unwanted inhibition of immune functions, more specific therapeutic approaches are of great interest, and Paul et al., that's the first author in our group, provide evidence that targeting the free fatty acid Phe2 A TLR4 mediated inflammation could improve glucose homeostasis in type 2 diabetes without affecting immune function. So next, talking briefly about macrophage polarization in adipose tissue, one of the previous speakers commented on it, that adipose tissue is polarized and infiltrated with M1 pro-inflammatory macrophages. What we have shown recently in our JBC paper is that M1 macrophages, they don't actually infiltrate the adipose tissue to start with. When you have unsafe storage of fat in the adipose tissue, what happens is you have polarization of M2 to M1 macrophages under the influence of Phe2 A. And this Phe2 A, we have for the first time demonstrated that it's not necessary that it has to come from the circulation. Inflamed adipocytes themselves can produce Phe2 A, which leads to macrophage polarization and transformation of anti-inflammatory M2 to pro-inflammatory M1 macrophages that gives rise to insulin resistance. So this work of ours got published recently in the Journal of Biological Chemistry. Adipocyte fetchwin A contributes to macrophage migration into adipose tissue and polarization of macrophages. Then at a clinical level, in a recent publication in Diabetic Medicine, we have shown that people with pre-diabetes in the highest quartile of fetchwin A level have a high concentration of IL-1 beta. I'm almost coming to the end of my presentation. Well, so we have anti-inflammatory approaches to treat patients with type 2 diabetes, and people are trying to block IL-1 receptor, IL-1 beta antagonists. They are using NF-kappa B inhibition, TNF-alpha antagonism to tackle inflammation-induced insulin resistance and type 2 diabetes. So to summarize, ladies and gentlemen, obesity-associated inflammation leads to insulin resistance, which is a pro-inflammatory state. Anti-inflammatory approaches therefore hold great promise, and our recent work shows that targeting fetchwin A and selenoprotein B may provide novel approaches to treat insulin resistance. Those who are more interested to know about the fetchwin A story, they may go through this recent review article published by our group in Endocrine Practice, Pro-Inflammatory and Anti-Inflammatory Attributes of fetchwin A. So with that, I end my presentation. Thank you for listening.